whatever. Okay, so on Wednesday we were talking about absorption, but really digestion and absorption, and we were talking about like how each molecule or each macromolecule type gets digested and absorbed. So just a quick, like one sentence description of each of these. So basically carbohydrates get chopped up by your saliva and pancreatic juice. There's amylase in both of those. The end products of amylase are disaccharides and limit dextrins, which are like small sugars, but not monosaccharides. So in order to actually absorb a carbohydrate, we're going to use enzymes on the cell walls that are going to finalize the breaking down of our carbohydrates. And we also have transporters that transport them in with sodium. So here we can see maltase breaking down maltose into glucose, and then glucose getting transported in with sodium. Proteins also have an enzyme that breaks them down called proteases. A couple different kinds. Endopeptidases split like internal bonds. Exopeptidases take amino acids off the ends of chains. All proteases are stored as zymogens, which are like inactive proteases in a bag somewhere in the cell. They don't get released until later. Okay, so proteins get digested starting in the stomach. An important protease, pepsin, is activated by acid in the stomach, so that starts breaking up our proteins. Protein digestion continues in the small intestine. Some of these inactive proteases, trypsinogen, chymotrypsinogen, and procarboxypeptidase, get released into the small intestine. They get activated partially using enzymes on the border of the small intestine itself, your enterokinase. And once they're activated, they can finish breaking down proteins either into really small chains or into amino acids, which can then get absorbed across the membrane, pretty much the same way glucose does. So they get absorbed usually using sodium or some other type of transport across. Regardless of what size the protein is when it gets absorbed into the cell, it gets broken down into amino acids before it gets put into the plasma. So both proteins and carbohydrates have some polar properties. Really, the important thing about both carbohydrates and proteins is that they're both water soluble. And lipids are weird because lipids are a nonpolar group. So we have to do like some extra steps in order to break down lipids. And we'll see that the absorption of lipid across the cells is actually super simple because of these nonpolar properties. So that's going to look different from the absorption of carbohydrates and proteins as well. Okay, so when we consume lipids, they basically form these big fat droplets because they're in a medium containing water and they're nonpolar, which means they're hydrophobic, which means they're oil and water, right? Like they don't mix well. So basically we're going to need to break down these fat globules, these droplets, into small enough sections that we get enough surface area that the enzymes can actually get out the lipids and actually break them down. So the enzyme that we're interested in when we're trying to break down a lipid are various lipases. So lipases are secreted from the pancreas. So they go out of the pancreas through the sphincter of Oddi into the duodenum. And remember that that's where bile comes out as well. And they can only act on molecules near the edge of a fat droplet. So they're going to have to work in conjunction with bile salts. So bile is also coming out of that same sphincter into the duodenum, but it's coming from the liver or from the gallbladder, which is like a storage organ for extra bile. And the bile salts are going to make our droplets smaller, and then the lipases are going to attack like the outside of these smaller fat droplets. So the word for what the bile salts are actually doing is emulsification. So you use emulsifiers just to make something nonpolar and polar mix well. So to mix oil and water, or if you're making like a salad dressing or something and you don't want to have like little clumps of oil in it, you'd try to add some type of thing that's an emulsifier. A lot of packaged foods have like I think soy lecithin or something is an emulsifier that's used in food. But in your body, you have bile salts. So bile salts are amphipathic molecules. And we've seen that word way, way back when we were talking about cell membranes. So our phospholipids were amphipathic molecules. Our bile salts are also amphipathic. So they have this non-polar part, which is made basically out of cholesterol. So here we see a steroid, which is a type of lipid, right? Cholesterol. But they also have these extra little polar groups that get added. And these polar groups allow the bile salts to interact with water. And then the lipid part, this cholesterol piece, can interact with the lipid that you've consumed, which really we're thinking about triglycerides because that's like 90% of the lipid material that's going through your GI tract. So these are going to work to break down our big globules into little globules because they can kind of like get in there and separate them and have like little bits that are touching our fat droplet, but also parts that can touch the outside water. Okay. So here we have our fat globule. It's in water. In order to break it down into smaller droplets, we add these bile salts. So coming from basically our liver, now we have our little small fat droplets. Okay. So this is the point at which we're going to start digesting them using our lipases. So in your slides, just note, I changed the number here because I can't figure out how you would get 3 fatty acids out of a triglyceride. I'm going to explain what a monoglyceride is in order to make sense why I changed it. So triglyceride, right, is a glycerol backbone with 3 fatty acid chains coming off of it. What a lipase does, what the pancreatic lipases do, is they break off fatty acids from the glycerol. But when they do that, they actually leave 1 fatty acid attached to the glycerol. And that's what a monoglyceride is that glycerol with just 1 fatty acid on it. So I think this number has to be 2 rather than 3 because one of those fatty acids is still in the monoglyceride. But regardless, our lipases are breaking down our triglycerides into smaller components that are going to be able to basically dissolve across cell walls. So when they break them down, we don't see in this image them interacting with the cell wall yet, or the cell membrane yet, but we'll see that in a second. But we can see that we have a triglyceride here, but we have some smaller fatty acids that we're starting to break off. So we do that with a lipase. So some of those during the process of breaking down are going to get absorbed into the cells like kind of immediately, but others are going to stick around in the lumen. And what they're going to do is form something called a micelle. A micelle is kind of like a fat droplet. It's a collection of lipids in like a little bubble. So you're going to have both kind of free fatty acids and these little clumps in micelles. And basically, as you absorb more of the fatty acids into your cells, you're able to kind of chip away at the micelles and get more and more particles out of them. So now let's take a look at the actual cell. So we had our fat globule, we broke it down with bile salts. So what we're looking at here is we have our fat droplet, and we can see that there are some bile salts still attached to it. We can see that there are some phospholipids in here. We have some triglycerides. Lipase comes in from the pancreas, and what it does is it's grabbing our triglycerides, right, from the edge of our fat droplet. So here you can see that it's grabbed on one of our triglycerides, which we know is a triglyceride because we can see the 3 fatty acids. And then it breaks it down. So it breaks it down into either just pulling off fatty acid chains on its own, or a monoglyceride. So that still has that glycerol backbone that was holding them together before, and 1 fatty acid chain kind of left on it. So those are basically the 2 types of things we can get when we break down our triglyceride. Some of them are going directly into the cell. So here we see an absorptive cell, so a cell that can absorb stuff, lining the lumen of our GI tract. And because fatty acids are nonpolar, we remember that like basically the reason we needed channels and all sorts of proteins in the membrane of a cell was because polar molecules and charged ions for that matter can't get across a cell membrane because it has like that double layer of fatty acid tails when the phospholipids face each other, basically. Like when we line up and form that bilayer, that phospholipid bilayer, there's a big non-polar region that a lot of molecules can't get across. So anything that's water soluble, basically, can't really get across the cell membrane. But fatty acids are not water soluble, and they're small enough, key additional point, I guess, to get across the cell membrane. So like a triglyceride is nonpolar, but it's kind of big, so it can't squeeze between phospholipids. But a fatty acid is small enough that it can fit between the phospholipids. And since it's still nonpolar, it doesn't get like blocked by that big nonpolar region in the middle of the membrane. So it can just passively diffuse across the epithelium. Okay, so here we see one fatty acid just doing that, just going straight across. And here we see that some of our other fatty acids are in a micelle. So this is our micelle. It's like a little clump of fatty acids. You see that there are some other types of lipids here too. So there are some phospholipids involved in this micelle, whatever. As these free fatty acids in the lumen get absorbed and there aren't any more, the micelles kind of start to break up and release fatty acids, which then get absorbed in the same way. Okay, so this is just simple diffusion, nothing active happening. It's just happening down gradients like you would expect. So once the fatty acids are inside the epithelial cell, some other stuff is going to happen. So they're not going to go directly across the basolateral membrane and into your blood. They have like their own pathway. So one thing that the lipids may do is that they may enter the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, one of our cell organelles, and what happens to our fatty acids there is basically that they can reform those triglycerides. So they can reform their larger shape now that they're inside the cell. Some other lipids get made here in the smooth ER as well using those fatty acids. Another thing that's going to happen inside the epithelial cell is that lipids can enter the Golgi apparatus or the Golgi body, whatever you want to call it. And they can be packaged into something called a chylomicron. So chylomicron is a type of lipoprotein, which is to say that it's just like a combination of a lot of lipid stuff and a little bit of protein. It's like a larger type of molecule. And chylomicrons are interesting to us partially because when you're measuring like blood cholesterol and stuff, one of the things you're actually measuring is certain chylomicrons of certain density. We're not going to get into the specifics of that, but there's a section on that in your textbook if you're interested. Okay, so some of these fatty acids and lipids are being turned into chylomicrons in the Golgi, and then they're going to get secreted by exocytosis. So they're going to get packaged into a vesicle. That vesicle is going to merge with the cell membrane. It's going to spit out our chylomicrons. Our chylomicrons are then in the interstitial fluid, but they're pretty big, like clumps of lipid and protein. And unlike our carbohydrates and proteins that we've digested and absorbed previously, the chylomicrons don't go into the plasma. So our carbohydrates and our normal proteins, the amino acids that we're absorbing, they're small enough that they can get across the layers or the capillary walls, I should say. So our carbohydrates and our amino acids from our proteins are going into your blood and they're going to be in your plasma. But our lipids that we're absorbing from food, our chylomicrons, they actually first go into the lymphatic system. So I think it's in the previous PowerPoint. So we remember basically that we have like these little prongs coming off off the inside of our GI tract, and they have blood flow to them, right? So they had like a little arterial going to them, a little venule coming out of them. They also had a branch of the lymphatic system that we called a lacteal. So the chylomicrons are going into those lacteals, which are part of the lymphatic system. And basically, the lacteals have bigger spaces between the cells, so the chylomicrons can get in there. The chylomicrons do eventually end up in your blood. Like, you need lipids to potentially break down for energy. A lot of them are going to get stored in adipose tissue as well. So they have to have a way to get there. But basically, your lymphatic system just eventually dumps into your cardiovascular system. So that's how they eventually get there. But when they're first absorbed, they're going through the lymphatics. Okay. So this kind of shows us what's going on with that. So we have our monoglycerides and our fatty acids. You see that this fatty acid is going by simple diffusion just across our cell membrane. It's going into our smooth endoplasmic reticulum. It's turning it into some type of bigger lipid. Oh, here it's turning it into a triglyceride. That triglycerides may be going to our Golgi apparatus, which is going to turn it into a chylomicron. So here's our chylomicron, which involves a little bit of protein as well. We can see it's in a vesicle, it's packaged, it's leaving this cell through exocytosis. It can't get into the blood. Well, that's okay, because it can get into a lacteal. So now it's in our lymphatic system and can go into our circulatory system once the lymphatics dump into the circulatory system. Okay. After our lipids are all digested and absorbed, we're actually going to reuse the bile salts. So recall that the bile salts were just breaking down our big fat globule into our smaller fat droplets. So that was mostly happening in the duodenum. By the time we get to the ileum, so the end of our small intestine, those bile salts are going to get absorbed into the capillaries. Remember that there's a hepatic portal system. So all our blood flow pretty much has to go back through the liver before it goes to the heart. So the bile salts head to the liver with the venous blood, and then the liver steals them and puts them back in the bile. So now we see the bile salt going back through the bile and getting pushed back into the duodenum. So we don't need to make too many new bile salts. We can just reuse the ones that we have. So these big macromolecules are not the only things we have to absorb from food. We also need to think about vitamins. So that people take vitamins every day. Hopefully you get a lot of vitamins in your diet. If you have deficiency of certain vitamins and minerals, it causes health problems. Different vitamins and minerals are going to be absorbed slightly different ways depending on their chemical properties. Some of your vitamins are fat soluble. So fat soluble vitamins include vitamins A, D, E, and K. These fat soluble vitamins, just like lipids, so they're basically going to be nonpolar, just like the triglycerides we just talked about. And they're going to be able to be absorbed basically directly across the cell membrane, just like we saw those little fatty acids getting across the cell membrane. So fat soluble vitamins get absorbed with the lipids. They can dissolve in lipid droplets. They can participate in micelles. Sometimes they're going to end up in those chylomicrons. Then they're going to get often actually like stored in your adipose tissue, so like in fat tissue with the lipids that you're otherwise storing. So sometimes when you're thinking about like taking your vitamins or eating vitamins, it's important to be careful about which ones you're like taking supplements for, because a fat soluble vitamin, because it interacts with lipids in this way, is actually going to stay in your body much longer, basically because it's not going to be like peed out with water when your kidneys like filter blood and stuff, because it's going to get stored with the lipids. Water soluble vitamins require special transport proteins. So water soluble vitamins can't get across the cell membrane on their own, so they're going to need help. So sometimes with water soluble vitamins, right, you'll hit kind of a transport maximum, right? If you don't have enough transport protein on the cell membrane, then it kind of doesn't matter how much vitamin you're taking. It's just going to basically, yeah, like not permanently get absorbed in your body because you can't get it all the way through. So vitamin B12 is an example of a water soluble vitamin. In addition to requiring special transport proteins, vitamin B12 can only be absorbed if it's bound to intrinsic factor. Intrinsic factor is something that's made by the parietal cells in the stomach. So you can basically only absorb a certain amount of vitamin B12 because it's reliant on these other proteins, these other things that you need in order to absorb it. So our minerals that we're interested in absorbing are basically ions. So we're interested in sodium, chloride, and potassium. Sodium and chloride get absorbed similar ways. So one of the ways that sodium is going to get absorbed is that it's just going to get dragged along with water. So as your cells absorb water, sodium just kind of hitches a ride. So that's one thing that can happen with sodium. The other thing that can happen is that it can be actively absorbed. And in the jejunum, the ileum, and the colon, there are active transporters for sodium. So your body is actively absorbing it, which shouldn't really come as a surprise because we've seen lots of sodium-potassium pumps, for example. So we know that sodium is something that cells do like to kind of actively control. Chloride ions aren't actively absorbed per se. Sodium has a positive charge, right? So as you absorb sodium, it pulls that positive charge with it because sodium is positively charged. So chloride just kind of follows because the negative charge on a chloride ion is attracted to that positive charge on the sodium ion. So it's going to go where sodium goes, and it can often kind of go between cells. So we've seen chloride before move by paracellular transport, so kind of getting through like leaky junctions between cells. Potassium is passively absorbed, mostly. So mostly potassium is just going to move passively across cell membranes. Another thing that's going to happen with potassium, though, is that potassium is going to get actually secreted into the GI tract when luminal concentrations, so the amount of potassium like in your GI tract, gets really low. So like if you have diarrhea, one of the things that's happening is that you have a lot of water moving through your GI tract. So you have a very dilute GI contents. You have a very dilute luminal concentration of pretty much everything, including potassium. So that's like a situation in which you would secrete potassium into the GI tract. So that's potentially going to mess, it's going to mess with like the potassium concentration in your blood, basically. Unsurprisingly, your GI tract is also processing bicarbonate. So we've spent a while on pH balance of the plasma. One of the places we're getting bicarbonate from is from the food we eat. So it's going to get absorbed. It's also going to get secreted because we need to control the pH of the GI tract itself. The place it's getting absorbed is mainly the jejunum. So bicarbonate ions are passively absorbed there. In the ileum and the colon, we're going to be secreting bicarbonate ions. And one of the reasons for that is there's a system where we can trade bicarbonate for chloride ions. So that is a way that we can get more chloride ions in the cell as well. We're also interested in absorbing calcium. Calcium is important for skeletal health, right? You build your bones out of calcium largely. You have like basically calcium crystals that make up the hard part of your bone. So we're gonna have to absorb that from your food as well. This is happening more like when you actually need calcium. So this is kind of modulated by the body's actual needs. When we are absorbing calcium because your body needs it, we're absorbing it in the duodenum and the jejunum, and we're doing it actively. The way calcium gets into a cell is that there are proteins on the apical membranes of some cells. So specifically on the brush border, which you remember is just that like kind of swirly part where we have a lot of surface area facing the lumen. So there are some proteins on the brush border, and they can bind calcium. When calcium binds to these brush border proteins, it gets transported into epithelial cells. At that point, the cells can actively pump the calcium into your plasma across the basolateral membrane using a pump. Vitamin D is also kind of involved in this process. It helps you absorb more calcium. Iron is another mineral that's super important to absorb. Important because we need it in order to have functioning hemoglobin, functioning red blood cells. We can end up anemic if we don't have enough iron. So iron needs to get into cells using transferrin. So we've seen transferrin before a little bit when we were talking about hemoglobin itself. So transferrin is secreted by enterocytes, so just like cells in your intestine on those walls, and transferrin binds iron. It's called transferrin because it like transfers iron, and like fer is like a root word that means iron. So transferrin binds onto iron, and then this transferrin iron complex can move through the system. So the transferrin ion complex binds to receptors on cells lining the intestine, and it can then be pulled into the cell through endocytosis. So we call this receptor-mediated endocytosis, just because like it had to bind a receptor so that the cell knows it's there, and then that like triggers endocytosis, and that's how it's getting absorbed. So it's not like moving through a protein, it actually has to have the cell membrane move to scoop it in. Some iron just gets stored in the enterocytes in the first place, and when we store it in the form of ferritin. Some of it we're putting into the blood. When we transport iron in the blood, we need to bind it again to transferrin. So we have other transferrin molecules basically in the plasma that are ready and willing to pull iron along with them. Water also gets absorbed in your GI tract. And this is important because even though you don't like have to drink that much water in a day, typical intake is about 2 liters. All these secretions, all this like bile juice, pancreatic juice, gastric juice, have a lot of water in them. So you actually secrete about 7 liters of water into your own GI tract every day. So you're taking in 2 liters, but you're also funneling a ton of water into the GI tract in order to get all these enzymes and stuff in there. So you need to reabsorb all that water so you don't get super, super dehydrated super quick. So water absorption is passive. It basically just follows solutes by osmosis. So as we're seeing these other things get absorbed across the membranes, basically, so like sodium, right? As these things get absorbed into cells, right, it increases their osmolarity, so water gets attracted to these concentrations of the other things that we're absorbing. Okay, and we will do these later. We're gonna move briefly into the next PowerPoint so that we actually get to the end of this unit by the end of next week. Okay, so the final part of the GI system that we want to talk about is basically how stuff gets controlled, a little bit more about secretion, and a little bit about movement of stuff in the GI tract. So I mean, like, specifically, we'll get there, actually. What I want to talk about today are our reflex pathways and some of how hormones are controlling digestive function. So we said before that basically the gut has its whole own kind of nervous system. That is true. And that's partially going to be involved in how absorption, secretion, how all this stuff is controlled. So we also do, though, have input from your central nervous system, right? Like every time I look at a picture of a donut, I want a donut, and sometimes my mouth starts to water. You can tell that your brain is involved in how you're ready to process food, right? Because salivation, creating saliva, that's like you're producing amylase ready to break down those carbohydrates. So we're gonna see that depending on what hormones we're talking about, and depending on what kind of like reflexes we're talking about, we're going to either involve the central nervous system, or we're gonna involve nerves in our stomach itself, or we're gonna just like involve the cells of our stomach and our intestines. So sometimes we refer to these as different like phases of gastrointestinal control. So it's the cephalic phase if we're thinking about stimuli that originate in the head. And by the head, we really mean your sensory organs, right? And your brain. So if you're just thinking about how much you want lunch, like I am right now, that's part of the cephalic phase. So sometimes you can get hungry just by thinking about food. You also actually have reflexes that start producing these different enzymes that we need to break down our different types of molecules as you actually taste food, even sometimes as you swallow food. That's going to help, that's going to kick off some of these nerves to produce different things. Smell also produces a reaction. So those are what we call our long reflexes. If we're seeing like secretion in response to any of these stimuli that aren't in the GI tract itself, that's a long reflex. Sometimes we're going to have stimuli directly in the stomach itself or in the intestines themselves that are going to create different hormones to be released, basically. So like how like physically full the stomach is would create a reaction, something like that. So some of these are going to be short reflexes if it's just directly controlled. Sometimes we're going to have long reflexes because they're going to like go back to, they're going to send a signal to the CNS, which would then send a signal back. So if we're talking about a stimulus that originates in the stomach, we call that the gastric phase. Or if we're thinking about a stimulus that starts in the intestines, we call that the intestinal phase. So these are really just labeling where the stimulus is. Cephalic phase, head; gastric phase, stomach; intestinal phase, intestines. So since we're kind of talking about regulation of stuff, we want to think a little bit about what we're regulating. So some of the things that we're going to be regulating are saliva secretion. So like when I see a donut, my mouth starts to water. That's one of these reflexes, right? So that's one of the things that we're thinking about. That's really what we're interested in when we're talking about these different gastrointestinal control systems. we're really talking about making the stuff that's going to be breaking down our food. So we'll be thinking about saliva secretion, so salivating. We're thinking about making acid and pepsinogen in the stomach, so like to break down proteins. Think about pancreatic juice and bile, so to break down lipids. And then we'll also think about rates of fluid movement through the GI tract. Starting with the top, so we're starting with saliva. Yeah, because that's the first thing your food's basically going to encounter. So when you eat food, the taste and texture of that food stimulate receptors in your mouth. So some of these are responding to like the physical touch of the food. So those are mechanoreceptors. And some of them are reacting to chemicals in the food. So those are the chemoreceptors. But both of them are going to send those signals to your brain, basically. So in the medulla, like in your brainstem, there's a salivary center. So it's like a processing center, an integration center that controls, it's going to control parts of your autonomic nervous system that are going to go back to the mouth and affect saliva production. So you're going to get this stimulus from in your mouth, going to go to these receptors, going to go to the medulla, to the salivary center, and then the salivary center is going to send out some type of autonomic signal. So we remember that hopefully that the autonomic nervous system, that's your sympathetics and your parasympathetics. So your sympathetics and your parasympathetics actually both make saliva, but they make different types of saliva. Side note here is that sight and smell also affect the salivary center, but those signals are not coming from your mouth, right? They're coming from your eyes or from your nose, depending on which we're talking about, but they're going to the same place. They're going to the same salivary center in the medulla. They're going to have the same kind of effects. So when we stimulate salivation, if we're stimulating the parasympathetic system, so parasympathetic activation, remember our parasympathetic autonomic nervous system is the rest and digest part. So when we stimulate the parasympathetics, we actually make watery saliva. When we stimulate the sympathetics, we make protein-rich, pretty viscous saliva, which, funny enough, kind of makes your mouth feel dry. Another place that we're going to see secretion kind of regulated is in the stomach. So here what we're looking at is acid and pepsinogen secretion in the stomach. And this is the pathway that acid's being made, basically what we're looking at here. So when we secrete acid into the stomach, we're really doing that basically like when we want to break down food. It's not useful to have a stomach full of acid for no reason. Kind of the point of the acid is to break down food. So when we want more acid in the stomach, what we're going to do is take CO2, H2O, we're going to use carbonic anhydrase like before, and eventually we're making hydrogen ions and bicarbonate, right? So on the basolateral membrane, that bicarbonate that we just made is going to get sent to your plasma, so that might affect your blood pH. And at the same time, we use the same protein to trade bicarbonate for chloride ions. So we pull in chloride ions into the cell. Then both the chloride ions and the hydrogen ions that we produced, basically made them from CO2, right, get secreted into the stomach. So they're now in those gastric juices mulling around in your stomach, and hopefully they're helping you break down like some proteins, right? They're gonna denature proteins so that you can start to break them down, specifically using pepsin, which we're gonna get from the pepsinogen being secreted by the stomach as well. So both acid and pepsinogen secretion are basically regulated like in sync. And this is good and useful because in order to get pepsin from pepsinogen, right, like in order to get the active form of that molecule, it needs to be activated by acids. So it just makes sense to secrete them at the same time in reaction to the same stimuli. And those stimuli are your parasympathetic nervous system. So your autonomics can come down to your stomach and be like, hey, secrete stuff. But we also have some more kind of local factors. So gastrin is a hormone that basically tells you to secrete acid, secrete pepsinogen, and digest stuff. And we also have super local factors. So histamine, and not just like any histamine, but like histamine produced by the stomach lining itself affects acid secretion. So if you have the cells of the stomach lining producing histamine, this would be like a paracrine sort of thing going on. So basically, they just go to the neighboring cells, send this paracrine signal to secrete acid and pepsinogen. So those are getting secreted together. We can also secrete, acid and pepsinogen, basically, through a cephalic phase regulation. So if you see food, or taste food, or smell food, or chew food, or swallow food, your brain knows that it's going to have to digest stuff. So if we get these stimuli, basically this triggers your parasympathetic nervous system to go tell the stomach to increase acid and pepsinogen secretion. The parasympathetic nervous system also stimulates gastrin secretion. So I'm going to scroll back for a sec. So remember that both parasympathetics and gastrin increase acid secretion and pepsinogen secretion. So these cephalic phase stimuli do like two parts of that, right? So we have our parasympathetic nervous system, and that has a direct effect on the stomach and affects secretion. And then the parasympathetic nervous system also stimulates gastrin secretion, which then goes and affects the stomach as well. So if we wanted to look at a flow chart of how this works, we see our input, right? So our initial stimulus would be the sight, the smell, the taste of food, or chewing and swallowing that food. It goes to your brain. Your brain ups the parasympathetic activity, so that branch of the autonomic nervous system. That goes to your stomach. Your stomach contains G cells. These G cells secrete gastrin. Gastrin gets into your plasma, and then that gastrin, so I'm following this sort of right-hand pathway here, and then that gastrin affects the parietal and chief cells and tells them to secrete acid and pepsinogen. Parasympathetic activity also goes just straight directly to your stomach, directly to those parietal and chief cells, and tells them to secrete acid and pepsinogen. So we're hopefully getting like a big response in response to actual eating. And I think we're going to stop there for now, and we'll get into the gastric phase regulation of the secretion on Monday. So I'll go check what's going on with everybody's grades now, so that will hopefully be less stressful. I'm sorry about that, guys.